DNA is the blueprint of life. It carries all vital information of life. But how this information gets translated into proteins is the mystery which had kept the scientists waiting for a very long time. The mystery was unfolded when the nature's genetic code was understood by the best brains in the field of genetics. When genetic code was designed, many mysteries relevant to human life were solved. Actually, a genetic code is an interface between inert genetic information and living organisms, as it plays a fundamental role in defining the process of evolution. In our lesson today, we will explore different aspects of genetic code.
Hello students, I welcome you all to today's lesson. We all know that Watson and Crick discovered that DNA is in the shape of a double helix. If you remember this double helix model of DNA, it has vertical twisted strands made up of phosphate and sugar. The base pairs link to form the inner ladder-like rungs that hold the outside strands together. Identifying the shape of DNA was a major breakthrough in genetic research, for which Watson, Crick, and Wilkins won the Nobel Prize in 1962. Before we start with the topic, let us briefly brush up our previous knowledge. We all know that during replication, one segment of DNA is copied to form another segment of DNA. The DNA transcribes to mRNA through transcription, which further translates to proteins through translation. These proteins are actually functional units of life. पर DNA ਵਿੱਚ present information proteins ਵਿੱਚ ਕਿਵੇਂ translate ਹੁੰਦੀ ਹੈ? ਇਹ mystery 1960 ਵਿੱਚ solve ਹੋਈ ਜਦੋਂ genetic code decode ਹੋਏ। ਅੱਜ ਦੇ lesson ਵਿੱਚ ਅਸੀਂ genetic code ਬਾਰੇ ਜਾਣਕਾਰੀ ਪ੍ਰਾਪਤ ਕਰਾਂਗੇ। ਪਰ genetic code ਨੂੰ ਸ਼ੁਰੂ ਕਰਨ ਤੋਂ ਪਹਿਲਾਂ ਅੱਜ ਦੇ learning objectives ਤੇ ਨਜ਼ਰ ਪਾ ਲਈਏ।
Upon completion of this class, students will be able to discuss significance of genetic code, describe genetic code and its function, list properties of genetic code.
ਮਨੁੱਖੀ ਸਰੀਰ ਵਿੱਚ ਕਈ ਤਰ੍ਹਾਂ ਦੇ ਪ੍ਰੋਟੀਨਸ ਦੀ ਲੋੜ ਹੁੰਦੀ ਹੈ। ਇਹ ਸਾਡੇ ਸੈਲਸ ਵਿੱਚ ਮੌਜੂਦ ਜੈਨੇਟਿਕ ਮਟੀਰੀਅਲ DNA ਹੀ ਹਨ ਜੋ ਕਿ ਸਾਰੀਆਂ ਪ੍ਰੋਟੀਨਸ ਨੂੰ ਪ੍ਰੋਡਿਊਸ ਕਰਨ ਲਈ ਲੋੜੀਂਦੀ ਇਨਫੋਰਮੇਸ਼ਨ ਪ੍ਰੋਵਾਈਡ ਕਰਦੇ ਹਨ। ਪ੍ਰੋਟੀਨ ਦੇ ਸਮੋਲੈਸਟ ਯੂਨਿਟ ਅਮੀਨੋ ਐਸਿਡ ਹਨ। ਇਹ ਪ੍ਰੋਟੀਨਸ ਸਾਡੇ ਸਰੀਰ ਵਿੱਚ ਸਟੋਰਡ ਜੈਨੇਟਿਕ ਇਨਫੋਰਮੇਸ਼ਨ ਰਾਹੀਂ ਸਿੰਥੇਸਾਈਜ਼ ਹੁੰਦੇ ਹਨ। This genetic information is represented by a three-letter word called codon. These three letters present in each codon are basically nitrogenous bases. There are 4 bases for 20 amino acids. This was a great puzzle for the scientists working on the subject. It was George Gamvo, a physicist, who worked on this puzzle. The puzzle was that how could these 4 bases code for 20 amino acids? It was reasoned that if a single nucleotide coded for one amino acid, then only 4 amino acids could be coded for. Alternatively, if 2 nucleotides specified one amino acid, then there could be a maximum number of 4 into 4, that is 16 possible different codes. If, however, 3 nucleotide code for one amino acid, then there would be 4 into 4 into 4, that is 64 possible different codes, enough to account for all the 20 naturally occurring amino acids. Dear students, these 64 combinations were enough to code for 20 amino acids. It is very well known that every amino acid is coded by at least one nucleotide, triplet or codon, and that some codons function as instructions for the initiation and termination of translation. It was a difficult task to prove that codon is a triplet. Sri Hargobind Khurana developed a chemical method for synthesizing RNA molecules with defined combinations of bases. But finally, Marshall Nirenberg and his colleagues tried to find a specific relationship between the nucleotide sequence and amino acid sequence. How did they find it? Just see.
Marshall Nirenberg and Heinrich Mattei conducted experiment by using mRNA composed of uracil only. Means all the codons were reading UUU or triple U. The protein thus produced was phenylalanine. What does it prove? Very clear. It means UUU codes for phenylalanine.
After this, a series of experiments were carried out by using synthetic mRNA consisting of alternating bases. By logical analysis of the end products, that is, various amino acids, it was possible to assign specific codons to these amino acids. This relationship eventually led to the complete designing of 64 codons in the form of genetic code. So what is genetic code? The genetic code is the set of rules by which information is encoded in genetic material, which further gets translated into proteins or amino acid sequences by living cells. The code defines a mapping between trinucleotide sequences called codons and amino acids. Now let us define genetic code.
Genetic code. The genetic code is the set of rules by which information encoded in genetic material, which is DNA or RNA sequences, is translated into proteins or amino acid sequences by living cells. The code defines a mapping between trinucleotide sequences called codons and amino acids. A triplet codon in a nucleic acid sequence usually specifies a single amino acid. As we know that the alphabets in the RNA molecule contains 4 letters, that is A, U, C, G, where A stands for adenine, U stands for uracil, C stands for cytosine, and G stands for guanine. To construct a word in the RNA language, that is codon, 3 of these letters are grouped together. An example of such a codon is ACG. In fact, every RNA word or codon corresponds to single amino acid. These codons are placed after each other in the RNA molecule to construct a message, a RNA sequence. This message will later be read by the protein producing machinery in the body. These codons and their correlation with the amino acids in a protein sequence define the genetic code. Now let us have a look at the table showing the genetic code. The 64 codons are divided into the 16 families, in which each codon begins with the same two bases. Means out of 3, first 2 bases will be the same in one family. For example, in this family, you can see the first 2 bases are common. Means 2 out of 3 bases in these 4 codons of one family are same. With the number of codons exceeding the number of amino acids, several codons can code for the same amino acid. For example, in this family, you see all 4 codons are coding for the same amino acid, that is leucine. 61 codons code for amino acids and 3 codons do not code for any amino acid. They function as stop codons. These are UAA, UAG and UGA, which do not code for any amino acid but instead signal the end of the protein chain. There is one start codon, AUG. AUG codon has dual functions. It codes for methionine and it also acts as an initiator codon to start protein synthesis.
Well, almost the same codons are assigned to the same amino acids, and there are the same stop and start codons in almost every plant, animal, and microorganism. Let me now describe the various properties of genetic code. All codons consist of 3 nucleotides. The code is degenerate. Out of 64 codons, 61 codons code for specific amino acids. One of the codons, AUG, codes for methionine, may also act as start signal. The 3 codons UAA, UAG and UGA are stop signals. Let us see and understand.
Properties of genetic code. The code is triplet, unpunctuated and non-overlapping. It became clear that 3 bases are required to specify each amino acid. There are no gaps between codons and these codons do not overlap. The code is degenerate. Most amino acids are coded by more than one codon. In fact, only methionine and tryptophan are coded by a single codon. One amino acid may be coded by more than one codon. For instance, serine is encoded by 6 codon, that is UCU, UCC, UCA, UCG, AGU and AGC. Glycine by 4, that is GGU, GGC, GGA and GGG. And lysine by 2, AAA and AAG. The genetic code is unambiguous. In general, no codon encodes more than one amino acid. The exceptions so far are AUG, UGA and UAG. There are 3 stop codons: UAA, UAG and UGA. there is one start codon, AUG. However, note that GUG and UUG are occasionally found as start codons. The genetic code is universal. Nirenberg and his colleagues did many experiments to find the universality in codes of bacteria and humans. Experiments with toads and guinea pigs showed that the code was almost universal. At a stage, Nirenberg looked out of the window of his office and said that the tree and the squirrel and himself would have turned out to be so similar biologically.
Wobble hypothesis. Did you notice one thing? In most cases, sufficient coding is performed by the first two bases, and the third base seems to play only a minor role. For example, the 4 codons that specify glycine are GGU, GGC, GGA and GGG. All start with GG. According to the wobble hypothesis, only the first two positions of a triplet codon on mRNA have a precise pairing with the bases of the tRNA anticodon. The pairing of the third position bases of the codon may be ambiguous and varies according to the nucleotide present in this position. Thus, a single tRNA type is able to recognize 2 or more codons differing only in the third base. The anticodon UCG of serine tRNA recognizes 2 codons, AGC and AGU. The bonding between UCG and AGC follows the usual Watson-Crick pairing pattern. In UCG-AGU pairing, however, hydrogen bonding takes place between G and U. This is a departure from the usual Watson-Crick pairing mechanism, where G pairs with C and A pairs with U. Such interaction between the third bases is referred to as wobble pairing. There are 3 terms: code, codon and anticodon. How do they differ? Although I know most of you are very well acquainted with all these terms, let me explain you once again. A code. Code here means the genetic code, which is the set of rules by which a gene is translated into a functional protein. These are the rules by which the base sequences of DNA are translated into the amino acid sequences of proteins. As you have already seen that the genetic code contains 64 codons. A codon is a sequence of just 3 successive nitrogenous bases. If it is of DNA, it will be formed of adenine, guanine, cytosine and thymine. But if a codon is of RNA, bases can be adenine, guanine, cytosine and uracil. Function of codon is that it specifies synthesis of a particular amino acid. Genetic codon also act as a signal to start and stop transcription or translation. In fact, a codon is the smallest unit of the genetic code. An anticodon is a sequence of 3 successive nitrogenous bases at one end of tRNA molecule. An anticodon recognizes and binds with a particular complementary codon on an mRNA molecule. Let us now summarize what we have learned so far. The central dogma of molecular biology suggests that the sequence of nucleotide in DNA specifies the order of amino acids in a polypeptide. ਭਾਵ molecular biology ਵਿੱਚ central dogma ਦੱਸਦਾ ਹੈ ਕਿ DNA ਵਿਚਲੇ ਨਿਊਕਲੀਓਟਾਈਡ ਦਾ ਸੀਕੁਐਂਸ ਹੀ ਪੋਲੀਪੈਪਟਾਈਡ ਦੇ ਅਮੀਨੋ ਐਸਿਡਸ ਦੇ ਆਰਡਰ ਨੂੰ ਡਿਸਾਈਡ ਕਰਦਾ ਹੈ। ਪਹਿਲਾਂ ਇਹ ਮੰਨਿਆ ਜਾਂਦਾ ਸੀ ਕਿ ਪ੍ਰੋਟੀਨ ਬਣਾਉਣ ਵਾਲੇ 20 ਅਮੀਨੋ ਐਸਿਡਸ ਵਿੱਚ ਹਰ ਇੱਕ ਦਾ ਕੋਡ ਹੁੰਦਾ ਹੈ। ਸਨ 1916 ਵਿੱਚ ਮਾਰਸ਼ਲ ਨਿਰਨਬਰਗ ਅਤੇ ਹੈਨਰਿਚ ਮੈਥੇਈ ਨੇ ਇੱਕ ਐਕਸਪੈਰੀਮੈਂਟ ਰਾਹੀਂ ਜੈਨੇਟਿਕ ਕੋਡ ਦੀ ਗੁੱਝੀ ਪਹੇਲੀ ਨੂੰ ਹੱਲ ਕੀਤਾ। The genetic code is the set of rules by which information encoded in genetic material is translated into proteins by living cells. The genetic code is a triplet code with codons of 3 bases coding for a specific amino acid. Genetic code उन्ना नियमां दी कुंजी है जिन्हां राही genetic material विचली encoded information living cells ਵਿੱਚ proteins ਦੇ ਰੂਪ ਵਿੱਚ translate ਹੁੰਦੀ ਹੈ। Genetic code ਇੱਕ triplet code ਹੁੰਦਾ ਹੈ ਜਿਸ ਵਿੱਚ ਇੱਕ specific amino acid ਲਈ 3 bases ਮਿਲਕੇ ਇੱਕ codon ਬਣਾਉਂਦੇ ਹਨ। Genetic codes ਦੀਆਂ ਕਈ ਵਿਸ਼ੇਸ਼ਤਾਵਾਂ ਹਨ ਜਿਨ੍ਹਾਂ ਨੂੰ ਇੱਕ ਵਾਰ ਫਿਰ ਦੁਹਰਾ ਲੈਂਦੇ ਹਾਂ। The codon is triplet. Out of 64 codons, 61 codons code for amino acids and 3 codons do not code for any amino acid. I hope you remember the 3 stop codons. They are UAA, UAG and UGA. Genetic code is unambiguous and specific. Can you name the amino acids coded by single triplet codons? AUG codes for methionine and UGG codes for tryptophan. The genetic code is degenerate, means most amino acids have more than one codon like leucine, serine and arginine. These all have 6 different codons. What are such codes known as? These are known as degenerate codes. The codes are universal. For example, UUU codes for phenylalanine right from bacteria to humans. ਭਾਵੇਂ ਮਨੁੱਖ ਹੋਵੇ ਜਾਂ ਜੀਵ ਜੰਤੂ, UUU code phenylalanine ਨੂੰ ਹੀ represent ਕਰੇਗਾ. There is one codon known as AUG, which has dual functions. It acts as initiator and codes for methionine also. ਮੈਨੂੰ ਉਮੀਦ ਹੈ ਕਿ ਤੁਹਾਨੂੰ genetic code ਨਾਲ ਸੰਬੰਧਿਤ ਸਾਰੇ ਗੁੱਝੇ ਭੇਦ ਸਮਝ ਆ ਗਏ ਹੋਣਗੇ। ਆਓ ਕੁਝ ਸਵਾਲ ਪੁੱਛ ਕੇ ਤੁਹਾਡੀ ਅੰਡਰਸਟੈਂਡਿੰਗ ਵੀ ਚੈੱਕ ਕਰ ਲਈਏ। With the help of the table given below, answer the following questions. First question. If mRNA has base sequence like CUC, AAG, UGC, UUC. The first question is, what series of amino acids are coded by this base sequence? The options are serine, tyrosine, arginine, glycine. Second option, valine, asparagine, proline, histidine. Next option, leucine, lysine, cysteine, phenylalanine. And the last option, proline, glutamic acid, leucine, valine. And the answer is leucine, lysine, cysteine, phenylalanine are the amino acids which get coded by CUC, AAG, UGC, UUC present on mRNA. Next question. For the same mRNA sequence CUC, AAG, UGC, UUC, name the anticodons present on tRNA. The options are GAG, UUC, ACG, AAG. Next option, GAG, TTC, ACG, AAG. Next option, CUC, GAA, CGU, CUU. Last option, CUU, CGU, GAA, CUC. And the answer is anticodons present on tRNA will be GAG, UUC, ACG, AAG. Next question is, as codon versus mRNA, find a similar relation in the given options. The first option is anticodon versus tRNA, triplets versus DNA, rRNA versus ribosomes, or all of the above. The answer is all of the above options are correct. My next question is, the codon AUG, which codes for the amino acid methionine, also serves as a start codon, a stop codon, a promoter, or none of the above. Answer, AUG also acts as a start codon. My next question is, the codons UAA, UAG and UGA all code for arginine, threonine, phenylalanine or stop codons. The answer is the codons UAA, UAG and UGA all code for stop codons. The next question is, we all know that the genetic code is almost universal in living organisms. This is based on the fact that all organisms, the options are evolutionary related, are genetically identical, have the same sequence of anticodons, or none of the above. The answer is because all organisms are evolutionarily related, therefore genetic code is universal. The next question is, anticodons are found on rRNA or mRNA or DNA or tRNA. The answer is anticodons are found on tRNA. My next question is, complete the following sentence. The entire genetic code consists of dash amino acids and dash codons. The options are 20-20, 20-64, 30-60 or 30-72. The correct answer is the entire genetic code consists of 20 amino acids and 64 codons. My next question is, complete the following sentence. A genetic code requires at least 20 different code words, one for each dash. The answer is, a genetic code requires at least 20 different code words, one for each amino acid. Next question. The genetic code is considered degenerate because the options are the code originated from bacterial genomes, DNA is unstable, or degenerate is the name of the person who deciphered the code, or more than one nucleotide triplet codes for the same amino acid. The correct option is the genetic code is considered degenerate because more than one nucleotide triplet codes for the same amino acid. Hope I have made a successful attempt to clear your concepts regarding genetic code. Thanks for your cooperation and attention. Looking forward to the next class. See you then. Thanks.